Hello, this is an introduction to microbiology tutorial presented by the SchrDbb. Reference Lippincott Illustrated Review of Microbiology by Cynthia Cornilesson, Bruce Fisher, and Richard Harvey. So, in this tutorial, we're going to visualize introduction to microbiology. So, we start by the overview. In the overview, according to Lippincott Illustrated Review of Microbiology, we have microorganisms are usually found in ecosystem, and then there exists a certain association that occur between microorganisms, and that association can be called mutualism. You have symbiosis. Symbiosis is just an association between two microorganisms. Now, when in that association, when the two organisms, both of them gain, it's called mutualism. But when one gain and the other do not gain, it's called commensalism. But even if one gain, the other gain, in commensalism, there must be a fact that the other who do not gain is not losing when the other one who is gaining is gaining. Is it clear? Now, in microbiology, what is the typical aspect in this chapter, in this subject matter, is the fact that the microorganisms are existing in relationship, or the living organisms are existing in relationship, but one organism is more living on the other. Is it clear? When one organism lives on the other, that one is now called parasitism. Is it clear? So parasitism is when one microorganism is gaining, while the other organism, who is the host, is called the host, is losing. Is it clear? That form of association or relationship is called parasitism. And now you need to know that before you have this infectious disease, there must be an initiating factor, which is colonization. And colonization is defined as the proliferation of microorganisms at the site of inoculation without any clinical symptoms to be found. Is it clear? That is inoculation. Now, you need to know that there are two main forms of pathogen that exist. You have prokaryotic pathogen and you have eukaryotic pathogens, and we are going to give the characteristics of each of them. So in this diagram now, we have some possible outcome following exposure to microorganism. So you need to know that at the first place, when a microorganism who is infectious is colonizing, colonization it first starts with contamination, and contamination is the presence of a microorganism which is not actively dividing on the inoculation site. Now the second thing is colonization. So now the microorganism now actively divide. These ones here are the cocci, they are circular in shape, while these ones here are the rods, they are like rectangular in shape. Now the next one now is that either the organism can become part of the resident flora, or the organism can be eliminated by the host defense mechanism. The organisms are going to be eliminated by a host defense mechanism, provided the organism try to invade the normal barrier of the organism. So like the normal flora, like in the oral mucosa, in the intestinal tract, in the vagina of the woman, all of them, they have normal mucosa, and the microorganism and the immune system is not reacting against those microorganisms because they are not impeding the normal integrity of the barrier on which they are placed. Is it clear? But now certain organisms will have this pathogenic ability where they have virulence factors such that they can penetrate the body and impede the normal functioning of the host. When they try to impede the normal function of the host, now that you have immune system try to defend yourself against this type of pathogenic organism. Now provided that now you have, so either it can become a normal flora or it can cause an infection. So when it causes an infection, you can have either a carrier state, that you have no clinical symptoms, maybe apparent, or the second thing is that the organism may be eliminated still by the host, and the third is that you can have pathogenicity, which can result now to an infectious disease. Is it clear? When infectious disease means that you can transmit that disease from one person to another. Is it clear? Now, the next thing are the different characteristics. Because I said there are two main types of cell types. We have prokaryotic cell types and we have eukaryotic cell type. The difference between each of them can be seen on this table. The chromosomes of the prokaryotic cell type is usually single and circular, while eukaryotic cell type is multiple. And the form of the chromosome in eukaryotic cell type exists like in pairs of sister chromatid. Now the next is the nucleus. So it can either exist as a chromatin thread, or the chromosome, when it is in a division form, can exist in a pair of chromatid. Now the next is the nucleus. The nucleus, we have no nuclear envelope or nucleoli with prokaryotic cells, while in eukaryotic cells you have membrane-bound nucleoli present. Now with the next one is membrane-bound organelles. Here you have no membrane-bound organelles like with phospholipids membrane-bound organelles. There are no. While here they are present. Example, you have the mitochondria, we have the endoplasmic reticulum, which consists of the rough and the smooth endoplasmic reticulum. Here the cell wall is usually present and it contains peptidoglycans. Now here cell wall is present, but it does not contain peptidoglycans. The cell walls of the plants contain cellulose. The cell walls of the fungi contain chitins. It is only the cell wall of bacteria, which are prokaryotic, which contain peptidoglycans. And these cells which contain peptidoglycans are gram-positive cell walls. Now the next one, we have plasma membranes. Plasma membranes at the level of the prokaryotic cell is made up of no carbohydrates and most lack sterols like cholesterols. While here there is the presence of carbohydrates and sterols, which can be the sterols, that is the cholesterol is helping in the flexibility of the membrane, and the carbohydrate is helping as a receptor on the membrane. The ribosome that exists at the level of prokaryotic cells are the 70S ribosome, while the ribosome that exists in eukaryotic cells are 80S ribosome. But you need to know that the mitochondria is usually considered as a bacteria which is in deep mutualistic activity with the eukaryotic cell. Is it clear? So it means that and this mitochondria contain 70S ribosome. So the question that I can ask you is, does eukaryotic cell do not contain 70S ribosome? The answer is no. Eukaryotic cell contain both 80S and 70S ribosome, because the 70S ribosome are present inside the mitochondrion, while the 80S ribosome is present inside the cytoplasm of the eukaryotic cells. Now the average size of these cells is usually smaller, so they are 0.2 to 2 mm in diameter, and these they are usually larger, that's 100 to 10 to 100 mm in diameter. Is it clear? So those are the different aspects of this microorganism. Now the next one, you have now inside the prokaryotic cell there are two main cell different. We have the typical bacteria and we have the atypical bacteria. Inside typical bacteria, you need to know that most bacteria have shape that can be described as either rods, spheres, or corkscrew appearance. Is it clear? So nearly all bacteria, with exception of the mycoplasma, have a rigid cell wall surrounding them. Now, in case of atypical bacteria, you need to know that atypical bacteria include organisms such as mycoplasma, chlamydia, rickettsia. Although they are prokaryotic cells, they are not actually having that typical characteristics of bacteria. Is it clear? So now, that is what you wanted to visualize here. Now the next one is now fungi. Inside the fungi, there are non-photosynthetic cells. Is it clear? And they are generally saprophytic and also eukaryotic. Now for protozoans are single cells non-photosynthetic eukaryotic organisms that come in various shapes and sizes. Those are protozoans. Now helminths. What are helminths? Helminths are just described as worms. Helminths are a group of worms that live as parasites. They are multicellular. In the case of helminths, unlike in case of protozoans, they are multicellular. They are eukaryotic. They have eukaryotic cells with complex body organization. Is it clear? And they are divided into cestodes, which are tapeworm, flukes, trematodes, roundworms, which are nematodes. Now the next one are viruses. The viruses are obligate intracellular parasites. That's why, since they are intracellular parasites, viruses are best said to, you can either say that a virus is living and a non-living aspect. Is it clear? And they do not have any cellular structure. It is only when they enter the virus, inside a living cell, that they acquire a metabolism. Is it clear? So viruses actually themselves, they are not living. So that's, in summary, now you can visualize the relative size of organisms. You need to know that the viruses are the smallest organisms, and they are here. Now we have what are called prions or proteins. Prions actually are certain protein types which are capable of giving a certain immune response, but they themselves, those proteins, do not cause any, they don't have any metabolism. So those ones here are called prions. They are capable of causing an immune response, but the prions actually are not living as compared to viruses. So viruses are larger. Then we have microorganisms here. Here, these microorganisms are the atypical bacteria. Consists of the mycoplasma, the chlamydia, the rickettsia. Is it clear? And they are very small. Then we have the size of the mitochondria. So see that the size of the mitochondria is even larger than the size of those rickettsia. They are not easily seen. That's why you cannot visualize a patient that is affected with chlamydia just by microscopy. There are typical tests which are used for chlamydia. Is it clear? Like the culturing of chlamydia and other sensitive tests that can be used for chlamydia. Now, for most bacteria, you need to know that most bacteria have the size of mitochondria. That's why the mitochondria, there is this exclusive theory of mutualism that exists, that says that's the way that a mitochondria is even a bacteria that is doing a mutualism with eukaryotic cells. Is it clear? Now we have the size of eukaryotic cells here, and then lastly, these are the size of large protozoans, and these are the size of worms. Don't forget that you can pause and then you can visualize the order of the size of each microorganism. And these are the only microorganisms that can visualize with electron microscope: viruses, prions, proteins, and small molecules. While these other organisms can be visualized with light microscope, but though they are going to be very minute on microscopy. Now the next thing is organizing the microorganism. Is it clear? So when you want to do organization of microorganism, you need to know that what in this textbook the authors have done the two-colored graphic formats. So you have hierarchical organization, you have list of important bacteria and viruses. So let's visualize the hierarchical classification of clinically important bacteria according to 6 distinguishing characteristics. So these are medically important bacteria. Now the medically important bacteria, we have one that lack cell wall. We have the second one, we have rigid cell wall, and the third one is the one which are flexible cell wall. So that is the classification based on the cell wall properties. Is it clear? So the classification based on the cell wall properties, we have lacking cell wall, we have rigid cell wall, and we have flexible cell wall. Now, based on the morphology, you need to know that the one which are having the rigid cell wall are divided into two. We have simple unicellular and we have filamentous based on morphology. Now, based on the growth inside or outside the host, the simple unicellular can be divided into obligate intracellular parasites, and they can be divided into free living. Now, based on the gram staining property, the gram staining property is you just use a gram stain, and those gram stains are going to attach on either peptidoglycans or non-peptidoglycans molecule. Peptidoglycans molecule are going to result to gram-positive microorganisms. So when they are free living, either they can be simple unicellular filamentous like actinomyces. Actinomyces are filamentous, or they are simple unicellular. Now, in simple unicellular case, it can be divided into obligate intracellular parasites parasites. Usually, oblique intercellular parasites are atypical parasites like Ehrlichia, like Rickettsia. All that they have atypical characteristics, like even Ureaplasma and Chlamydia, is it clear? And Mycoplasma, all that are oblique intracellular parasites. Now, free-living organisms. The free-living organisms are divided into two: either the ones that have peptidoglycan cell wall, or the ones that do not have peptidoglycan cell walls. The ones that have peptidoglycan cell wall are called gram-positive, and others do not have, they are called gram-negative. That's based on the gram-staining properties. Now, based on the shape now, we have gram-positive can be divided into cocci, which are spherical in shape, can be divided into rods, which are rectangular in shape, and the gram-negative still can be divided into cocci and rods. Now, based on the oxygen tolerance, all these can be divided into two, which are aerobes and anaerobes. That's a simple hierarchical classification based on their different characteristics. Now, this is the classification now of medically important virus family. So when you want to classify 5 viruses, how do you do? Medically important viruses are divided into two. We have RNA viruses and we have DNA viruses. Now, for RNA viruses, it's divided into two now. We have double-stranded RNA viruses and we have the single-stranded RNA viruses. Is it clear? So it can be called icosahedral, and we have non-envelope. So these are viruses. They don't have any envelope. They are double-stranded. They can be single-stranded. Now for DNA viruses, they have single-stranded, non-envelope, and we have double-stranded. Is it clear? Now for the RNA virus, the single-stranded now is divided now into these different parts, while for the double-stranded DNA is divided into two, which is the non-enveloped and the envelope. Is it clear? So when you are double-stranded, either you can be envelope or non-envelope, and when you are single-stranded, you can also be envelope and non-envelope. So you have here a single-stranded can be negative strand helical. It's negative strand helical and envelopes, and why you have here positive strand. The question you want to ask me is, what is the meaning of a negative strand and a positive strand? Negative strand just means that the direction of the RNA do not follow that normal direction of DNA. You need to know that one. Normally, a DNA is said to be positive stranded when it is moving from a segment of a DNA is said to be positive stranded when it is moving in a 3' to 5' direction. Is it clear? So a segment of a DNA is said to be positive stranded when it's moving from a 3' to 5' direction. Now, when that DNA does a complementary replication to when it does a complementary binding with an RNA, it is going to produce an RNA in a 5' to a 3' direction. Is it clear? That's why any direction which is said to be from 5' to 3' is said to be considered as a positive strand, while any direction which is opposite to that normal direction, so from 5' to 3' on an RNA is said to be a positive strand, while a 3' to 5' direction on an RNA is said to be negative strand. Is it clear? So those are the different cases. And now the positive strand now is divided into two. You have the non-enveloped icosahedral. Icosahedral means that the envelope is just icosahedral. Is it clear? So non-stranded, and then the shape is icosahedral, and then we have the envelope. Now inside the envelope now we have the icosahedral and you have the helical. Is it clear? So those are the different aspects that you have to visualize here. Now this next diagram now is medically important bacteria discussed in this book. So you have different bacteria that you're going to visualize in the book. So the medically important bacteria, I'm not going to speak about that too much, but I'm going to explain all of them. So let's just visualize the name of the bacteria. We have Coxelia bruneti, we have Ehrlichia and Rickettsia. We said that all these bacteria are said to be atypical bacteria. We have Actinomyces, Arachnia, Mycobacterium, and all that. These ones here are also other bacteria. These are Staphylococci, Entococci, and all that in the chapter. We have Mycoplasma. We're going to see Hominis incognitus, pneumonia, and Ureaplasma urea lacticum is also a mycoplasma, and we're going to see all the different microbacteria in the chapter. For the viruses, these are the different classification of viruses. You need to know that what the single-stranded non-enveloped viruses. An example is Parvo B19. Now we have the double-stranded non-enveloped virus. It's the adenovirus, the BK virus, human papilloma virus, the GC virus. The double-stranded envelope virus. You have Epstein-Barr virus, hepatitis B, herpes simplex, human cytomegalo virus. You can visualize here. Now the single-stranded positive strand RNA virus. These ones here are DNA viruses, and these ones here are RNA viruses. So when this RNA virus, you have Coxsackie, you have ecovirus, you have enterovirus, hepatitis A, and all that. You have the single-stranded positive, you have all these viruses. You have the single-stranded negative strand, helical, and envelope. You have all these viruses: California encephalitis virus, Ebola virus, Hantan virus. You have the double-stranded icosahedral non-envelope virus. An example is rotavirus, and all these you are going to visualize in this chapter. Now the next one now is the normal chapter inside the introduction is normal flora. Now, normal flora, what is the normal flora? The human body continuously inhabited by many different microorganisms, mostly bacteria, but also fungi and other microorganisms. So it means that what? The normal flora are those microorganisms which are just living inside the body, in a relationship with the host, that is the human body. And that relationship can be either commensalism or mutualism. So that relationship is a symbiosis. Is it clear? Now, normal flora does not constitute what is called parasitism, where one is going to gain and the host is going to lose. No, there is nothing like that in normal flora. So for normal flora, either one gain and nobody lose, or no one gain. Is it clear? So there is no loss inside symbiosis with normal flora. So the next one is the human microbiome. So you need to know the different parts of the human bodies and the parts that are constituting of the different normal flora. Is it clear? So I'm going to speak about that. So here you need to know that the level of the skin is an example. The skin consists of Staphylococcus aureus, Staphylococcus epidermidis, and Streptococcus species at the level of the skin. And also on the skin we have the Corynebacterium species, we have the Propionibacterium acne. But Propionibacterium acne is usually associated with a disease called acne, because that disease cause acne is mostly associated with the infection Propionibacterium acne, but as a result of excess testosterone level in the body. Is it clear? Though Propionibacterium acne is the one causing, which is a normal flora of the skin causing acne, which is a disorder. That disease is usually as a result of excess level of androgens in the level in the body of the person who is being affected. Is it clear? So these are the things. So now when you have now a disruption of the normal skin, these microorganisms which were normally functioning as in a normal flora are going to disrupt that skin, and then they are going to cause an infection. Is it clear? So now we have the different microbiomes. So we said example of bacteria that inhabit now at the level of the conjunctival sac. You have gram-positive cocci such as the Staphylococcus aureus, Staphylococcus epidermidis, Streptococcus species. Also at the we have the gram-positive rods like the Corynebacterium species, Propionibacterium acne, and we have gram-negative cocci like Neisseria species, and we have Moraxella. These ones are going to inhabit at the level of the conjunctival sac. Is it clear? At the level of the eye. Now, these are the example of bacteria now that inhabit at the level of the mouth. So the one that inhabit at the level of the mouth, we have gram-positive cocci. In gram-positive, you have Staphylococcus species, you have Staphylococcus sanguinum in the mouth, sanguinus, you have Streptococcus mutans, and these Streptococcus mutans are usually the ones which are involved with the dental caries formation. Now the next one, you have gram-positive rot. You have Corynebacterium species, Propionibacterium acne, still located in the normal flora there. We have the gram-negative cocci, is the Neisseria species. You need to know that we have Neisseria species inside the mouth, mostly Neisseria gonorrhea. For people who are doing oral sex, is it clear? They are going to be present as a normal flora inside the mouth. Now the next one, you have gram-negative rots like Haemophilus species. Usually you may have even Haemophilus influenzae inside the mouth because you are already immunized against Haemophilus influenzae. But you need to know that there are different types of Haemophilus influenzae. Usually it's Haemophilus influenzae type B, which is the most pathogenic, and that one we are already immunized. And this one is not present as a normal flora. Usually you can have Haemophilus influenzae even type A or type C and other types. Usually those are the less pathogenic forms. Now you have anaerobic organisms, you have bacterial species still at the level of the mouth, you have Fusobacterium, you have Prevotella species. Now the spirochetes, you have the Borrelia, you have the Treponema. Is it clear? Now the spirochetes we said that there are three main forms of microorganisms based on their shapes. We said that they were cocci, they were rods, and they were corkscrew. They were even comma shape. And this one, the spirochetes, are the ones which are spiral in shape. Is this clear? So the microorganisms which are more spiral in shape are said to be the spirochete. And remember that all these were containing the cell wall. Is it clear? Even with the anaerobic microorganism, the spirochete also contains the cell wall. So spirochete, we have Borrelia, we have Treponema, and we have others like Actinomyces. Now the Actinomyces are the ones which are more filamentous, though they still contain the cell wall. They are filamentous. So these are the different microorganisms. Now the organisms that most often implicated in plaque formation are Streptococcus mutans, which are gelatinous glucans and constitute plaque material. So just those plaques are actually in French they are called tartre. So there are these things, there are these whitish things which are present at the level of the teeth. Is it clear? So when you have your teeth, you see inside your teeth you have whitish material which can be found at the junction of teeth. Those ones are called plaques, and the Streptococcus mutans, which is the same one that causes dental caries, are usually the cause of those plaque formation. Is it clear? So the next one, and after the mouth, we have example of bacteria that inhabit the gastrointestinal tract. You start with gram-positive cocci. At this level, usually staphylococci is now, you see that staphylococci was far present up here at the level of the mouth as this first species of the gram-positive cocci, but now inside the gastrointestinal tract, staphylococci is not still far up. The first one which are inside here are going to be the Enterococcus species, and then the Enterococcus. You need to know that the Enterococcus species are part of the Streptococci family. Is it clear? The Enterococcus species are part of the Streptococci family, because you need to know that what the Streptococci have different groups. You have group A, group B, group C, group D Streptococci. Is it clear? And then the Enterococcus family are example of group D Streptococci. Is it clear? Now we have Peptostreptococcus again. It's another example of the Streptococci family. Now before we have Staphylococcus species. Now Staphylococcus species is an example of Staphylococcus species. An example is a Staphylococcus, and then we have the Streptococcus viridans, and the Streptococcus viridans is an example of the group B bacteria. Now group B Streptococci. Now we have the gram-negative rods. Inside the gram-negative rods, you have the Enterobacter species, we have the Escherichia coli species, we have the Klebsiella species, we have the Proteus species, we have the Pseudomonas species. All these are the gram-negative rods. Now the anaerobic organism, we have the bacterial species, we have the Clostridium species, the Lactobacillus species. Now among these anaerobic organisms, now we have a clinical correlation is that Bacterial Vaginosis is the one that causes intra-abdominal abscesses, and it is the main one that is involved involved with peritonitis when you have a ruptured appendicitis or diverticulitis, or even a ruptured intestinal due to an intestinal trauma. And then other organisms that can still be present inside the normal flora, we have actinomyces. So now the next one is example of the one that inhabit now the vagina. So inside the vagina we have the gram-positive cocci, we have Staphylococcus species, we have gram-positive bacilli, we have the Corynebacterium species, and the main one here we have Lactobacillus. Now we need to know that what this flora, the level of the vagina, are also called the Dodeline flora, Dodelerlein, Dodeline flora. So those Dodeline flora, actually you need to know that what when you are asking for the culture of the vaginal flora, there is not only one single microorganism that is going to come as the culture. Is it clear? It is multiple microorganisms that are going to come as a culture. Is it clear? And those multiple microorganisms are called the Dodeline flora. Is it clear? So those microorganisms are usually, you need to know that this microorganism, which Dodeline flora, is mainly made up of Lactobacillus, and that's the most common organism that is in the normal vaginal flora, because that Lactobacillus produces an acidic pH that is not favorable for the growth of other microorganisms. Now we have gram-negative rods such as the Proteus, you have the Pseudomonas, you have the Gardnerella, you have the Klebsiella, and all those species, even the Streptococcus. Now the gram-negative rods, if the gram-negative rods are excessively high than those gram-positive bacteria like Lactobacillus species, that is going to be very bad, because the patient is going to be having an atypical vaginal flora. Is it clear? Normally this gram-positive bacilli, which is made up of Lactobacillus, should be having the highest proportion of the microorganism found there. But if these ones are higher, then it is highly sure that the patient is going to suffer from what is called a smelly vaginal discharge, fishy smelly vaginal discharge. Is it clear? And that is called vaginosis, bacterial vaginosis. So we have anaerobic organisms here. We have now bacterial species. Now the mycoplasma organism, we have mycoplasma species. Usually if this is going to be high, then the patient has a high sexually transmitted infection. And we have actinomyces species. So from here we have visualized this on that chapter. Then we now go to the next part of the chapter, which is called the pathogenicity of the organism. Now, inside the pathogenicity of the organism, we need to know that what there are different definitions that you should know in the overview. We have what is called subclinical. When you say when we mean a subclinical infection, is an infection that has no detectable symptoms. So you have an infection in you without any symptom. Example is asymptomatic gonorrhea. The next one, which usually occur in females. The next one is latent. So latency is an infection with the potential to become active at some times. Here, the patient do not have the microorganism inside the body. Is it clear? When you are saying that the infection is latent, it means that the microorganism is not actively dividing. Is it clear? That's why it's called latent. An example is the latent phase of syphilis, the latent phase of Mycobacterium tuberculosis. Is it clear? So syphilis in case of Treponema pallidum, and tuberculosis in the case of Mycobacterium tuberculosis. Now the next one is opportunistic. Opportunistic is an infection which is due to an organism that generally does not cause disease in the normal host. Is it clear? So it is because the immune system is compromised that microorganism now is going to cause now an opportunistic infection. Example, when you have HIV, or so when you have AIDS, you are going to have certain diseases like pneumocystis pneumonia caused by pneumocystis carini or pneumocystis jirovecii fungi, and that fungi is due to immune suppression. For that infection is due to immune suppression from HIV. Now the next one is primary. So when you are said to have a primary infection, a primary infection is an infection by organism that may be latent or later cause other disease manifestation. Actually, actually it's better to say it this way. When you have a primary infection, it means that you had the infection for the first time. So or you can consider a primary infection is an infection for the first time. That's the first case. And the secondary infection is usually infection for the second time. That's the first way you can visualize that definition. Primary infection is one when you have been infected for the first time. Secondary infection is when you are infected for a later time. Is it clear? Now that is it. Or you can still define primary as being the first stage of an infection and secondary as being the second stage of an infection. Is it clear? So those are the different ways you can visualize it. So infection by an organism that may become latent or later cause other disease manifestations. So the primary infection can either become latent, it can stop, or it can continue to a secondary phase. So example, we have Treponema pallidum. Now secondary infection. In secondary infection is the second time that you are infected. It can be a reactivation, or you are infected the second time. Is it clear? Or it can also be a second stage of an infection, as I have said. Either it can be second time you're infected, or it's a reactivation of a latent disease, or it's the second stage of a disease. Still, in the case of Treponema pallidum and Mycobacterium tuberculosis, even plague, you're going to see in the case of plague, which is caused by Yersinia pestis, we have the bulbous phase and we have the pneumatic phase of the plague that you're going to visualize based on primary and secondary infection. Now we have mixed. So it's when you have two or more bacteria infecting the same tissue. So mixed infection is that. Example is pelvic inflammatory disease, which can be due to Neisseria gonorrhea, Chlamydia trachomatis, and other microorganisms. Usually you need to know that pelvic inflammatory disease is affected by 3 major microorganisms. We have Neisseria gonorrhea, we have Chlamydia trachomatis, and we have anaerobes. Is it clear? That's why we have association of 3 main antibiotics, which is fluoroquinolone. Fluoroquinolone is going to, or a third generation cephalosporin, which is going to target the Neisseria gonorrhea. We have the second one, which is tetracycline, like doxycycline, which is going to target Chlamydia trachomatis. Or you can see, if you don't want to use a tetracycline, you can still use a macrolide, or you can still use a macrolide, which is like azithromycin, which is going to target Chlamydia trachomatis. And the third microorganism you use, the third antibiotic you use is a metronidazole and an imidazole. Example, metronidazole, which is going to target anaerobes. Is it clear? So those are the 3 major microorganisms which are involved in pelvic inflammatory disease. Now we have pyrogenic. So pyrogenic just means post formation. Is it clear? Example of post formation microorganism, we have staphylococci, we have streptococci, and staphylococci and streptococci are the ones which are forming spores. Is it clear? But the one that live inside the pores is not actually staphylococci and the streptococci. It is another microorganism type. Is it clear? So the staphylococci and the streptococci, they predispose to the formation of pores, but the one that live inside the pores typically are not the aerobes, because usually staphylococci and the streptococci are aerobes. It means that they need air, while inside the pores, inside an abscess, there is no blood vessel to transfer oxygen. So that's why inside the pores, usually there are anaerobes which live there for the maintenance of the pores. Is it clear? So that's why every time that there is an infection, there is an infection with pus formation, you must need to add an anti-anaerobic, example an imidazole, like in the case of metronidazole. Now the next one is fulminant. The definition of fulminant is that infection that occurs suddenly and intensely. Example is necrotizing fasciitis from Streptococcus pyogenes. So it's also called a flesh-eating bacteria, necrotizing fasciitis caused by Streptococcus pyogenes, flesh-eating bacteria. So that is what you're going to visualize. So the next one, we have the virulence factor. The virulence factors, so the other definition, so this diagram now is going to tell you how microorganisms have their different virulence factor. Is it clear? The virulence factor can just be defined as the factors that predisposes the microorganism for an infection in the host. Is it clear? So it first starts with the entry into the host with evasion of host primary defenses. So the microorganism evades the host primary defenses, enter the human body. It can enter through inhalation, through the oral route, through the urogenital aspect, and through the rectal aspect, or even through the skin. Is it clear? And even with different contact with vector transmission. Is it clear? Or even cuts. Now after that, you have now adhesion to the organism host cells. So now the cells, those microorganisms have adhesion methods. Is it clear? They can use their pili, which are these different filaments at the level of the bacteria. You have the pili, and the pili are these segments which are used for the communication between two bacteria. You can use their pili or any adhesion molecule to adhere on the surface, like the glycolipids of the microorganism host. Is it clear? And this part here is the flagella. Now the next one here, you have, after the adhesion of the root, the host is going to now enter through the cell junction, or it can pass through the cell, and then it's going to propagate now into the bloodstream. So that's what you have. This one is invasion of the host. Invasion of the host is when it enters through it passes the host barrier, like the mucous membrane. It passes into the bloodstream, or via all those different routes. After the invasion of the host, now it now goes into propagation, where the microorganism is going to be pumped to a different part of the body, or it can be propagated through the interstitial fluid of the body, or it can be propagated through the lymphatic circulation of the body. So any method there which can be propagated is called propagation. Now we have damage to the host by bacterial toxins. The bacteria can produce toxins, or they can damage the host by themselves. So this is the toxin mediated damage of the host, and this is the damage of the host by host immune. Even the host immune response can damage the cells, because when the immune cells identify that a cell is affected, it's going to destroy that cell. And then later you have either progression of the disease, or you have resolution of the disease. Here is going to divide more to cause a progression of the disease, and here bacteria is going to be eliminated by the immune response. Is it clear? So those are the different mechanisms. That's the pathogenesis, physiology of how mechanism of infectious processes. Now the next diagram here is going to show you the action of exotoxins. So in action of exotoxin, you need to know that in this diagram, ADP is adenosine diphosphate, ADPR is adenosine diphosphate ribose, and NAD plus is nicotinamide adenine dinucleotide. So it starts by number 1. So when you have action of exotoxin just means that what the toxins of the microorganism are secreted out of the bacteria, while endotoxin means that they are toxins which are only secreted when the cell wall of the bacteria is being ruptured. That's when the bacteria dies, and the endotoxin is going to be secreted. So for exotoxin, you have the membrane recognition of the portion of toxins bind to the cell membrane. So you have that the cell membrane, this is the toxin here, is going to bind to the cell membrane and it's going to enter. So after entering the cell, a subunit is going to dissociate and cleave and modify to the target molecule. So you have you have active subunit is going to enter into the cell, and you have these different processes. Is it clear? And then the toxin now is going to destroy the host microorganism. So you can visualize all this to visualize the different action of the exotoxin. So now this diagram here is going to telling you the Koch's postulate, because you need to know that what actually in the past it was thought that microorganism was produced from spirits, or were produced from different energies, were produced from different bad molecules and all that. So these were different theories that were given in the past. Is it clear? But now Koch, who is the founder of the Microbacterium organism, now postulated. Is it clear? So Koch postulated he gave 3 theorems that actually there is a microorganism that is causing diseases. Is it clear? It's stipulated that well, there is a microorganism that is going to cause a disease, and if that microorganism is if that microorganism is identified and is inoculated in another species, the patient is going to have the same clinical features like the initial patient. Is it clear? So those are the three main parts of the Koch postulate. The first thing is that microorganism is the one that causes a disease, and then if that microorganism is identified and also cultured and inoculated in another living organism, that living organism is going to have the same clinical features like the previous living organism. So this is a case here. So we have a sick mouse. Let's take a case where you have a sick mouse. Here the microorganism is identified and that microorganism is isolated. That was even the word I was looking for. So the microorganism is isolated. So when that microorganism is isolated, now it is now going to be inoculated in another living species, like that same living species, like the initial sick mouse. Now when it's inoculated, that species now is going to have again the same clinical feature after the microorganism is inoculated. Also, the isolated microorganism must cause the same original disease like the inoculated when inoculated into the susceptible animal. Is it clear? And the microorganism can still be re-isolated again in the experimental infected animal. Is it clear? So that's the Koch postulate. Very important theory inside microbiology. So that's telling you that there is actually the existence of microorganisms, and that those microorganisms are the ones that are involved with the clinical symptoms and diseases of the patient. So the next one is this image here, which shows you the types of viral pathogenesis at the cellular level. Is it clear? So viral infection that result in host cell death. So it's actually, you need to know that actually the host cell death is because the virus is split multiple times. Is it clear? So when the virus is split multiple times, it's now going to cause an expansion of the host cells, and that's going to cause in the breakage of the host cells. Is it clear? So viral infection results in a transformation of host cells. At the beginning, when you have viral infection, the virus is going to inoculate their genetic material. It can either be DNA or RNA. If it is RNA, RNA is going to be converted by a reverse transcriptase. Is it clear? The reverse transcriptase is just called reverse transcriptase because it is a reverse of transcription. Is it clear? So normally you convert from DNA to produce RNA, but if you convert from RNA to produce DNA, that's called a reverse transcription. So the enzyme is going to help you in causing that reverse transcription. Is going to be a reverse transcriptase, and that reverse transcriptase now can produce a DNA that is going to move now into the DNA of the host cell to change the host metabolism. Is it clear? That's the first method via which an RNA virus can result now to a change in the host metabolism. The second method is that what the RNA, you need to know that what RNA constitutes of the RNA can also be in a form of a messenger RNA. Is it clear? And that messenger RNA can directly go to the ribosomes and cause the production of proteins without passing through the nucleus or the DNA of the cell, the host cell. Is it clear? So those type of microorganisms that have RNA that can directly go to the ribosome and cause the protein production do not have reverse transcriptase for the production of the DNA. So they are going to just directly go as an RNA for the production of proteins at the level of the ribosomes. Is it clear? And now for DNA viruses, it's easy. The DNA is going to directly go at the level of the DNA of the host cells, and it's going to cause a change in the immune response, the change in the metabolism of the normal cells. So now we have viral infection that result in host cell fusion. So other viral infection can result to host cell fusion. Is it clear? So that's one. Is it clear? So these are the different so one is going to result to virus, is going to result to host cell death. Other one is going to result to transformation of the host cell metabolism. Other is going to result to host cell fusion. So because you have virions inside, you have viral proteins, and those viral protein causes the fusion of the different host cell to produce a giant multinucleate cell. And you have cytopathic effect. Here you have cell rounding, you have stainable viral protein, and you have cell disintegration. So all these are the type of viral pathogenesis at the level of at the cellular level. Now this diagram is going to show you examples of dissemination of viruses to secondary sites in the body. Is it clear? So how do viruses disseminate secondary sites in the body? Now we start with an infection. So we have body surface. It now go to the lymph nodes. Is it clear? So after the lymph nodes, it now enters into the blood. The blood can cause the passage of virus into the bone marrow, into the liver, into the spleen, and other blood vessels. Is it clear? So this one is called primary viremia. Primary viremia is when it has passed through the lymphatic, enter into the blood for the first time, and then go into the different tissues. Now secondary viremia is when it has entered the blood for the second time, the presence of viruses in the blood for the second time. Now the presence of viruses, so you need to know that when it enters the blood for the first time, it's going to contact the major organs like the bone marrow, the liver, the spleen, and the blood vessels. These cells are mostly the reticular endothelial cells, which are involved in the production of immune response. After the secondary, the primary viremia, we have now a secondary viremia. When the virus have passed the immune response, it can enter the blood again the second time. Now when it enters the blood for the second time, it's now going to enter the nasal cavity or oral mucosa, where it's going to cause infection. Can also go to cause infection inside the skin, can also cause infection inside the central nervous system, inside the lungs, and all the like. Is it clear? So after the secondary viremia, that now you have the disease. This disease that affect the nasal mucosa and all that is called rubella. This disease that affect the skin is called measles. This disease that affect the brain is called poliovirus, and this disease that affect the salivary that is called with the lungs is called cytomegalovirus. This is an example of case where you have a viral infection. You need to know that viral infection only comes after a secondary viremia, because the primary viremia is going to result to is going to enter first into the reticular endothelial tissues, which are going to try to remove the microorganism from the circulation. Now this is now the different modes of transmission inside of microorganisms. Transmission can either be done of microorganisms, can either be done horizontal transmission or vertical transmission. So let's start with vertical transmission. Vertical transmission is transmission from an infected mother to a non-infected child. So infected mother to a non-infected child can come via 3 routes. Is it clear? The first route is in utero, where you have a transplacental spread, meaning that what the virus or any other microorganism can penetrate through the placenta into the fetus. The second is during delivery. Is it clear? So this is the antepartum. Is it clear? This one is the intrapartum. During delivery, now blood can enter through the nostril to the different ligatures of the fetus, and then it enters into the body during delivery. And then the third is during the breast milk. So this is another form of vertical transmission. Those are the 3 main methods of vertical transmission to a child. Now the next is that some viruses are transmitted from mother to infants. Example are the torches infections. Not only viruses but microorganisms transmitted from mother to child is torches. T for toxoplasmosis, O for others, R for rubella, C for cytomegalo, H for herpes simplex, as you can visualize here. You have herpes simplex type 1 and type 2, human cytomegalo. You also have human immunodeficiency virus, HIV. You have rubella. Those are the mnemonics can just be crammed as torches infection. T for toxoplasmosis, O for others like in the varicella, HIV, R for rubella, C for cytomegalovirus, H for herpes simplex, E for Epstein-Barr virus, and S for syphilis. So all that was visualized. Now the next thing is diagnostic microbiology, still in the introduction. So in the introduction, you need to know that there are different diagnoses you put out in microbiology. So these are the laboratory techniques that are useful in diagnosis of microbiology. Here, number one, you can do direct microscopic visualization. This one is called just microscopy. The second one is just culture and sensitivity, when you do cultivation of and identification of microorganisms. You need to know that on culture, there are certain parts in which are produced by a specific microorganism. Is it clear? Certain microorganisms are going to produce circular pattern, others are going to produce rectangular pattern. There are different microorganisms that are going to produce different pattern on the culture, on the culture meal. Is it clear? The next one, you have detection of microbial agents. So you have other methods, detection of microbial antigen, and that example of detection of microbial antigen can occur by PCR or by the use of certain molecules. The next one, you have detection of microbial RNA and DNA, still by PCR. This one, detection of microbial antigen, example like Treponema pallidum hemagglutinin assay, which is used for the detection of Treponema pallidum. Treponema pallidum is actually an example of this case here. Here is PCR, and here you can have also detection of the host immune response, like when you want to detect antibodies. An example, you want to detect antibodies against rubella, immunoglobulins against rubella, you can use this host immune response antibody detection. So all these are the detection of microorganisms. The first one, you can do direct microscopy, culture and sensitivity. The third one is detection of the antigen, like Treponema pallidum hemagglutinin assay. Is it clear? Or you can also do the venereal disease research laboratory. So that can be also used for detection of the antigens. The next one is detection of the microbial RNA and DNA, mostly by PCR. And the next one is detection of the immunoglobulins of the host. So all these are the 5 main things that you have to detect when you want to do microbiology. The next thing now, we have the heat. So this diagram now is going to show the step of gram staining method. Is it clear? So in purple you have gram positive microorganism, while in red is gram negative microorganism, while the colorless, there are those ones that do not do any gram stain. So this is the preparation. So we have a heat fixed specimen to the slide. So you fix the specimen to the slide. We have flood slide with crystal violet solution. Is it clear? Allowed to act for 1 minute. Now you have to rinse, then flood the slide with iodine solution. The first thing is that when you do gram staining, you first heat the specimen on the slide. So fix the specimen by heat on the slide. So that's the first thing. After you flood the specimen with crystal violet solution on the slide. After flooding the patient with crystal violet solution on the slide, the next one is to rinse the slide. Then you flood the solution now with iodine. You flood the slide now with iodine solution and allow it still for another 1 minute, just like this case here. You put it iodine solution. Is it clear? So before acetone decolorization, that's the next step. So after you put, so the next one now is you need to know that one, before the acetone decolorization, all the organisms appear purple. That is gram positive. Is it clear? So it means that when you rinse, you put the iodine solution. So after putting the iodine solution, the organisms that are gram positive, is it clear, are going to appear purple. Before you go to the next step, all the organisms that you have placed now iodine solution are going to appear purple. Now the next case now is to rinse again the excess iodine here and then decolorize with acetone for approximately 5 seconds. In this case, here is for 1 minute and here is for 5 seconds. Is it clear? The time depends on the density of the specimen. So you wash the slide in water. After acetone decolorization, those organisms that were gram negative are no longer visible. Is it clear? Is it? So you need to know that all the gram are when you had iodine first here. So iodine is going to bind with the gram-positive bacteria only, and they are going to make the gram-positive bacteria they are going to make the gram-positive bacteria are going to appear purple. Now, after the acetone, you place now acetone here. After the acetone being placed, it's going to decolorize, is it clear? And now the gram-negative bacteria are no longer going to be visible. Is it clear? Now you apply again the sofranin counter stain for 30 seconds. Is it clear? You wash with water, and then the gram-negative bacteria are going to be visualized after the application. You have the gram-negative bacteria, you have the gram-positive bacteria. So all this is a step in order to visualize the gram stain. Let me go again once again. You first start by heat facing the microorganism specimen on the slide. After you apply crystal violet for 1 minute. The next thing is to rinse and then apply the iodine solution. After applying the iodine solution, now apply acetone solution to decolorize all the gram-negative bacteria. After decolorizing them, you apply sofranin for 30 seconds. After applying sofranin, now you are going to visualize 2 microorganisms. One microorganism is going to be purple. That one is going to be gram-positive. The one which are reddish are going to be gram-negative. While the one which are colorless here, they don't react with sofranin. Is it clear? And they are weak gram-positive. You need to know that in this diagram here, you had also purple color. They are going to be weak gram-positive, meaning that what they do, when they react with the iodine, they are still decolorized by acetone to become colorless. Those weak gram-positive are colorless here, and they are not considered as gram-positive. Is it clear? So these are examples of cases of Mycobacterium tuberculosis which is stained by acid-fast stain. This is another staining method. If it's not gram-positive stain, can be acid-fast stain, which is going to stain Mycobacterium tuberculosis. And for acid-fast stain, the stain that is mainly used is the Zellnersen stain. Now here is the India ink preparation now for Coccus neoformans in cerebrospinal fluid. These seed cells are identified by large transparent capsule that exclude the India ink particles. Now these are the fungi in unstained nasal sinus exudate made distinct from other materials such as cells with potassium hydroxide. So when you use potassium hydroxide stain, you can visualize the fungal hyphae. Is it clear? Now again, this is with anaerobic transport media containing a non-nutritive medium that retard the diffusion of oxygen after addition of the specimen. Microorganism may remain variable for up to 72 hours. It is important to use the airless syringe for liquid specimens such as pus rather than a swab and tube or to transport the medium. Actually, just this image is just telling you that what you have an anaerobic media. An anaerobic media is a media which do not contain air, and that media is very important because microorganisms are capable of surviving in that media for at least for 72 hours. And it is very important to take this media in consideration. Why? Because most microorganisms are opportunistic microorganisms. Is it clear? They are not the major microorganism involved inside the infection. So let's think about the fact that you are collecting a substrate. Let's say that you are collecting a substrate. After collecting a substrate, you have collected some of the Staphylococci aureus in microorganisms which were found at the level of the normal floor of the skin. Is it clear? So when connecting the substrate, you see that there is also going to be some of the Staphylococcus aureus with the typical pathogenic microorganism at the level of the specimen. So to prevent the growth of the Staphylococcus aureus, so that Staphylococcus aureus should not grow on the culture media, you should store that, you should collect that specimen in an anaerobic media. Is it clear? So when you should have to place that specimen in an anaerobic media. Why? Because the microorganism, even if they are aerobic, they are going to survive in that anaerobic media for at least 72 hours. Is it clear? So when they survive in that media for at least 72 hours, it's going to impede the growth of the non-major Staphylococcus aureus. Is it clear? So that transport media is going to be very important now for cultural media. So you have to prevent this. So the next one now is, this is an example of a cultural media. Is it clear? So you have a Marconi agar. So it's a lactose fermenting for gram-negative rots. So you need to know that there are certain different media which are good for different microorganisms. That's why it is good to specify the culture that you want on as a medical doctor on your request form. Is it clear? So this one is a lactose fermenting gram-negative rot. Only lactose fermenting gram-negative rot are going to grow inside the Marconi agar. For example, bacteria that ferment lactose produce acid that decrease the pH of this agar. Is it clear? And the acidity now causes the pH indicator in the agar to turn now pink. Is it clear? So now you have other tests which are commonly used to identify microorganisms. And the first test we had here, one test was just it was just culture and sensitivity. Now these tests now are other tests which can be used to identify common microorganisms. Example, we have the catalyst test. The catalyst test, you have bubbles indicate that you have production of oxygen gas by catalyst. Is it clear? You just place peroxide inside a solution containing the bacteria, and you're going to visualize bubbles of air, just meaning that that bacteria is going to be catalyst positive, and you need to know examples of bacteria which are catalyst positive. So these methods are not actually used more and more now because there are more sophisticated methods that can be used. But in case where you are in a far away center, you still use this method. The second method is oxidase test. For oxidase test, you're going to have a dark oxidized product that is going to result from cytochrome C oxidase activity. Actually, in oxidase test, you are going to trying to find if an organism is going to oxidize a substrate. Is it clear? So you are going to place a substrate in two parts. So these organisms which are going to be found here are going to produce a dark purple color, which means that they are oxidase positive. They are capable of oxidizing a material. While these other organisms are oxidase negative, there is no bubble. Is it clear? Here you are going to have bubble, so it's catalyst positive. You have no bubble, you have catalyst negative. Now the Uvs test is another test. Here is pH indicator in agar turn dark. So you just placing a agar, pH indicator in agar turn dark due to the production of ammonia by urease. So in urease positive organism, you have the production of ammonia, and that's going to make the agar to turn dark. So the urease positive. So when placing in a urease positive media, you place a certain substrate. After placing the substrate, now the one which are urease positive are going to turn dark red, while the one which are urease negative, the media is going to stay same color. The next one, you have the coagulase positive bacteria and the coagulase negative bacteria. So for coagulase positive bacteria, we are going to promote clotting of the sebum. Is it clear? So normally you need to know that there are certain enzymes like coagulase enzyme which are involved in blood clotting. Like example of Staphylococcus aureus, you need to know that Staphylococcus aureus is a microorganism which is involved mostly with blood clotting, can clot microorganism, and when it clots the microorganism can result to ischemia of certain parts. Like that's why coagulase positive microorganisms coagulase positive Staphylococcus aureus are very, very virulent microorganism. Is it clear? So when you have coagulase test, you can just take a blood agar and then you place the microorganism inside. If the blood has clotted, then it's going to be coagulase positive test. If the blood has not clotted, it's coagulase negative test. As easy as that. Now you have other tests again. You have the A is the Vtec Test card containing the test word, and you have the color of word changes with time. So you have the reagent of word. You can just place the reagent of word inside the different test tubes. Now the reagent of word, you have test inoculum enter the port and is distributed among 30 words, each containing a different biochemical substance, and you have the different time. So the color changes in the word result from metabolic activity of the microorganism. So you have the different. Now this one here is the rapid manual biochemical system of bacterial identification. Is it clear? So this is another manner to visualize the color of whorl. So is it clear? Different appearance of the upper and the lower part of the whorl indicate positive and negative ability of the bacterium to utilize each substrate. Is it clear? So this is another test which can be used in order to identify microorganisms, and this is the Vtec test. We're using the card, using a card with a test whorl stain. We just use the whorl reagent, and this test is called the Vtec test. Is it clear? The whorl reagent is you place the different specimen, and the different specimen are going to produce different colors based on the different microorganisms, based on the different substrate of that whorl reagent that they can react on. Is it clear? So there are different colors that are going to be produced by the different substrate, and those colors can be helpful now in the identification of the microorganism. Is it clear? So actually I'm not versed with this test, that's why I cannot tell you more, but you can just pause and then you can visualize on this diagram. Now you have the next one is complement fixation. Complement fixation now is another method now to visualize the microorganism. So here you are trying to fix the complement factor. Other cases is that you have immunoglobulin fixation. So a fixed amount of antigen is added to the test serum. Is it clear? If the antibody is present in the serum, the immune complex will form. Is it clear? So you add antigen in the serum. Now if you have antibody in that serum, it's going to react. Is it clear? So when you have the you have the already antibody, just add the antigens of that, is going to put the immune complex. So if you have serum with this one is serum without the antibody, so that you have added the antigen, and now this is serum with antibody. Now the next one is complement is then added to the mixture. You now add the complement factor to the mixture. If complexes are present, then it will fix the complement now to it. Is it clear? So here you have antigens without the antibodies here. So this test now is to try to identify the immunoglobulins. Just this is the complement fixation. I use the complement fixation method. So here this test would simply is going to have antigens without antibodies. You have inoculated antigens. Here you have antibodies in this test tube, that you have inoculated antibody for the production of immune complexes. Then you consider by the inoculation of complement factor. Here inoculation of complement factor is going to result in nothing. While here inoculation of complement factor is going to bind down to the upper terminal of the immunoglobulins, which are bound to produce the immune complex. Is it clear? Now the red blood cells and the agglutination amount and anti-erythrocyte antibodies are added. Is it clear? So you add the red blood cells inside. Is it clear? So you add the red blood cells inside. When you add the red blood cells inside, antibody against the red blood cells now are going to react. Is it clear? So if the complement has not been bound, anti-erythrocyte antibody plus the complement lyses the red blood cells. If the complement has bound by the immune complexes, there will be insufficient complement to lyse the red blood cells. Is it clear? So I hope you've just understood the principle. So in this one, that there was complement without the immune complexes, you see that the complement are not going to cause the lysis of the red blood cells. After adding the red blood cells, the complement are not going to cause the lysis of the red blood cells. So here you add the red blood cells and you add the agglutination factors, the agglutinins. Is it clear? You add both the red blood cells and the agglutinins. The agglutinins themselves are not the ones that are causing the lysis of the red blood cells. It is the complement which is causing the lysis of the red blood cells. Is it clear? It's actually the complement which is causing the lysis of the red blood cells. So you add both the red blood cells and the agglutinins. Example, you can add the anti-A antibody. So now, since the complement here has not bound with the immune complex, it's going to bind with those antibodies and those red blood cells to cause the lysis of the red blood cells in this first case. But in the second case, since he has bound, there is not going to be any complement factor to bind with the antibody and the red blood cells. So though there is antibody and the red blood cell, there is no complement factor to bind with it. So here there is no lysis of red blood cell. So is it clear? So in this test, if the blood become colorless, it means that the test is negative. But if the blood, the test tube is still reddish, it means that the test is positive. Complement fixation has occurred, and means that that specimen was containing antibodies of microorganism. The next one is another stage. So if you have a schematic representation of antigen agglutination latex bead, is it clear? So this is the bound antibodies. This is the latex bead. This is the antigen. So you can still bind. So the latex bead, the antigen you can bind to the antibodies here. You have particle agglutination. So particle agglutination, diagram A is going to be positive with positive latex bead, and this one is diagram B is negative latex bead. Is it clear? So those are the latex agglutination method of identifying microorganism. Now the principle of enzyme link immunosorbent assay is another method of identifying microorganism. So if you still want to identify antibodies, so antibody specific for an antigen of interest is bound to the cell wall of the plastic microliter, as you can visualize here. So we want to visualize the certain antigen. For the first case, complement fixation was used to visualize antibodies. Is it clear? But now in this case now is you are trying to visualize the antigen. So when you want to visualize antigen, you are going to use the ELISA, enzyme link immunosorbent assay. So antibodies here, if you use an antibody that is going to be linked to the substrate, to the plastic microliter, the serum now is placed. So if that serum now contains the antigen, so you have patient serum is inoculated in the wall, any antigen in the serum is going to bind now to the antibody of that wall. Is it clear? Now you wash the wall. Now after washing the wall to remove the unbound antigen, now you place now an enzyme here antibody. You place an enzyme antibody substrate. So you add again a second enzyme labeled antibody specific for the different epitopes. So you add it. After again you wash the wall, so the enzyme labeled antibody is added, and then you wash again the wall to remove the unbound antibody. Is it clear? So after washing now, you add now a product. So enzyme make color produced from added substrate intensity. So now there is a product at the terminal of an enzyme here. So that terminal of enzyme, you now add another substrate that the enzyme is going to act on. Is it clear? So if there is a conversion in color of that substrate that the enzyme act on, it means that there is a positive ELISA test. It means that the antigen here was actually found on in that first specimen. Is it clear? So this is the enzyme link immunosorbent assay. So these are the figures. So this figure now is going to show commercial nucleic acid amplification system for diagnosis of infectious disease. So you need to know that there is another test which is called the nucleic acid amplification testing, NAAT. Is it clear? And the nucleic acid amplification testing is just there for specific microorganism. An example of microorganism that is going to be visualized by nucleic acid amplification testing, we have Bacillus anthracis, Bordetella pertussis, Chlamydia trachomatis, cytomegalovirus, enterovirus, Epstein-Barr virus, hepatitis B, C, herpes simplex, human immunodeficiency virus, human papilloma virus, methicillin resistant Staphylococcus aureus, Mycobacterium tuberculosis, Neisseria gonorrhea, SARS coronavirus, which is severe. So we have vancomycin resistant enterococci virus, zoster, variola virus, and West Nile virus. All these are involved there. Now, this negro diagram is going to show you an outline of this diffusion method for determining the sensitivity of bacteria to antimicrobial agent. So it's another method of determining microorganism. So you use a sterile nutrient agar and use organism to be cultured. So this is how you want to culture microorganism. So sterile nutrient agar, use organism to be cultured. You inoculate, you use disc containing different antibiotics. So you place, so this is for culture and sensitivity. You place a disc here with an antibiotic, you place a disc here with another antibiotic, you place a disc here with another antibiotic, and you place a disc here with another antibiotic. Now incubate now for 24 to 48 hours. Antibiotic diffuse into the agar away from the disc. So you are going to see it's going to diffuse. So you have zone of inhibition of growth, you have zone of inhibition of growth and other. You need to know that what if there is a large zone of inhibition of growth, it means that the organism is going to be sensitive to that particular microorganism. Like here, you see to that particular antibiotic, like in this place here. So you see that the microorganism is going to grow all through the nutrient agar. Is it clear? A sterile nutrient agar is going to grow all through here, but it's not going to grow at this particular position where you have the disc here. Why? Because it's sensitive to both the antibiotic on this disc and the antibiotic on this disc. Is it clear? Why at this level where you have this antibiotic and this antibiotic here, the organism is resistant to those antimicrobial agents, to the antibiotics here and antibiotic here, because it grows at that level. Is it clear? Now this is the determination of minimal inhibitory concentration of antibiotics. So now you have tubes containing varying concentration of antibiotics are inoculated with test organism. Is it clear? So example, you have 64. So here you know the antibiotic which are used. Is it clear? Now you want to know the dose of the antibiotic to use for each microorganism. So you can use different tests with different concentration of microorganism with the inoculating agent. So you place here 64. This is the highest antibiotic concentration and this is the lowest antibiotic concentration. Here is 64 and here is just 0.5 concentration. So these are the relative antibiotic concentration. Now do growth of the microorganism now in a measured 24-hour inoculation incubation period. So after testing that, let's say that amoxicillin is the sensitive antibiotic to that organism, you put different concentration of amoxicillin in a test tube and then you make them culture. Is it clear? So if you see that there is no bacterial growth till 2, it means that 2 is the minimal dosage of amoxicillin that you have to use. Is it clear? And it means that if you use just 1 and 0.5, those are the minimal inhibitory. This 1, 2 here is called the minimal inhibitory concentration. It's the lowest concentration of antibiotic that is required in order to inhibit the bacterial growth. Is it clear? And that's the concentration that you have to use in the human body. So the next one is control no drugs. So this one is the effect of bactericidal and bacteriostatic drug on growth of bacteria in vitro. Is it clear? So this is the bactericidal drug. So the bactericidal drug here is going to decline the microorganism. Example of bactericidal drug, we have penicillin. It's going to decline microorganism. Is it clear? It's going to kill the microorganism and make sure that the microorganism die or eradicated. Now bacteriostatic prevent the growth of microorganism. Is it clear? It does not, it makes sure that the microorganisms do not continue proliferating. So those are bacteriostatic agents. Example is chloramphenicol, and this is a control where no drug is used. Is it clear? So from here you have visualized all the different parts. Now the last chapter under antibiotics, under the introduction of microbiology, is going to be vaccination and antimicrobial agents. So you need to know that this is the instance of vaccine preventable disease in the United States. So you see that tetanus is prevented with vaccine. You see that poliomyelitis has been prevented with vaccine. So tetanus has been reduced. These are the number of cases, yearly incidence. It has been reduced even to 10 cases per day per year. Poliomyelitis has been reduced even to 1 per year. Rubella, mumps, diphtheria, pertussis, and rabiola. So you see that these are the diseases. So circles here at the upper end of the arrow that indicate the incidence of disease when before the vaccines were available at this point here. While this arrow is going to show after the disease, when the vaccine has already been was already available, those were the cases. Now, these are the standard human immunoglobulins. Standard human immunoglobulins, hepatitis A or measles, used in individuals who have been exposed to the virus. You use immunoglobulins. Now, you have hyperimmune human immunoglobulins in the case of botulinism, in the case of diphtheria, in the case of hepatitis B, rabies, and all that. These are the immunoglobulins used for passive immunization. Those are the different cases you can use immunoglobulins. You can use hepatitis A or measles, can be used in botulinism, in diphtheria, in hepatitis B, in rabies, in tetanus, and in varicella zoster virus. Now, these are the different instances of microorganisms and the different bacteria that are going to attenuate. I'm not going to speak more on vaccination because I'm going to speak about a different tutorial on the different bacterial vaccines. Is it clear? So from here, I'm going to say thanks for your kind attention, and please don't forget to like and subscribe for our channel. Thanks very much.